Today we're going to talk about how bacteria reproduce. Now we have talked about reproduction before, and that we said there's basically two types. You either have sexual reproduction or asexual reproduction. And since we did that entire chapter on genetics, we talked about meiosis and mitosis, and so you're pretty familiar, I think, probably with how an organism is going to reproduce sexually, because you're going to have two parents that are going to each provide half of the genetic material to their offspring, and then the offspring are going to be different from the parents. In asexual reproduction, you have one parent and the offspring are going to be identical to the parent, unless, again, there's some sort of genetic mutation. Bacteria only reproduce asexually. And so we're going to talk first about how that process is going to happen. They reproduce by a method called binary fission. Fission and fusion are opposite words. And so fusion means that you stick two things together if they fuse together. Fission is when they come apart. They break apart from each other. So fission is breaking apart, and binary, the prefix bi means two, just like a bicycle has two wheels. So binary fission is a reproductive process where you're going to have one cell that's going to break apart into two new cells, okay? So if this is a bacteria, and this is a very simplified version of a bacteria, obviously, but here's a bacteria, and when it's getting ready to reproduce, its genetic material, it's going to attach itself in this ring to the cell wall. Then the bacteria is going to make a copy of that. You know, we've talked about how DNA makes a copy of itself, so you're familiar with how that process works. So I'm going to try to kind of see if I can show you how this works here. So this bacteria is then going to copy its genetic material, and it's attached very, very close to the place where the first piece is attached, but it's not in exactly the same place, it's just close. Then what's going to happen is that this bacteria is going to start to elongate, and it's going to make more and more of the plasma membrane as it goes down, and so it's going to start spreading apart like this until it eventually separates those two rings. So eventually what it looks like is once it has reproduced itself, and I've got that second ring of genetic material, it elongates. So then I have one ring of genetic material here and one ring of genetic material over here, and it separates itself. And then what's going to happen is those are going to pinch apart and separate so that each one has its own ring of genetic material in it. Okay? So that is binary fission. So basically you've got a way where it just is the genetic material reproduces, attaches to the cell membrane starts spreading apart, and then eventually the two pieces are going to split apart. So I have two new cells, and again, they're both identical to the parent because that was the only source of genetic material that they have. Now, there is a way, actually there are about 3 ways that bacteria can gain some genetic variation. So they can gain some variation in their DNA, but they do not do it through sexual reproduction. There's about 3 other ways that this can happen. The first way is by conjugation. Now, what conjugation means is that I've got one bacteria here, and here's its main ring of genetic material. So here's its DNA, the vast majority of it, but it also has a little piece of DNA down here, and this is called a plasmid. And a lot of times that plasmid is going to specify for a very specific trait within the bacteria. Then there's another bacteria over here who has the DNA, but it doesn't have the plasmid. And so we're not quite sure how all this works, but somehow this bacteria can sense that this one doesn't have this extra piece of DNA. And so because this is going to be beneficial for its survival or for its other traits, it wants to donate this additional genetic trait to the other one. So what it does, the next step of the process, is they build, so I've got this, I think I erased my plasmid over here. It wouldn't be good. So here's my two bacteria. This is the one with the plasmid. It makes a copy of the plasmid, and then it builds this conjugation tube in between the two bacteria. And the copy of the plasmid travels through the conjugation tube and into the second bacteria. So then, once that copy has transferred over through the conjugation tube, I then have two bacteria, and they each have that additional plasmid with the additional trait in there. Now, that's not sexual reproduction because in sexual reproduction, both parents are going to donate genetic material to the offspring. But that's not what happened. Here I just had one donor and one recipient, and they both end up with the new material, but it's not sexual reproduction because you don't have two parents. You just have a donor and you have a recipient. Okay? Now, how do I remember this term with this process? Well, I like to look at pieces of words and see if that can help me remember things. So in the word conjugation, I see the word jug. Well, what's a jug? A jug is something that, like if I have a jug of apple cider, it's the fall of the year here right now, and so if I have a jug of apple cider, I can take that apple cider and I can pour the apple cider into a cup so that I can drink the cider. So a jug is something that I use to pour with. Well, in conjugation, I think of how this bacteria kind of pours this DNA, this extra plasmid, this genetic material, into the other one. So that pouring of the DNA through the tube, through the conjugation tube, is how I remember that conjugation is what this particular process is. So hopefully that will help you to remember it. Okay, the next method of genetic recombination, and we say genetic recombination because, again, it's not asexual reproduction, but the genes have kind of been recombined in a way so that this bacteria has traits that it didn't have before. Transformation is going to do the same thing. Now, this one's kind of funky. I have a bacteria that has some DNA in it, and what happens is that this DNA dies, and when it dies, the cell wall all disintegrates, and then the DNA kind of starts to disintegrate also, and so then I have just these pieces of DNA just kind of floating around in the environment. So the cell walls have fallen apart, the contents flow out. Another bacteria that's still alive over here sees this, well, it doesn't see it, but it somehow senses that this DNA is there. It senses that the DNA would be beneficial to it if it took it in. And so probably by using phagocytosis, it's going to take these pieces of DNA into it. Remember how phagocytosis works. It kind of goes out and takes in these other pieces of bacteria. And so when it takes it in, it's going to take it in and create a plasmid out of it. Okay? So basically, in order for this to work, you have to have a bacteria to die. Its DNA is all kind of floating around out here. This sees a piece of it, it engulfs it, and then makes a plasmid within its own cell. Okay? Again, that's kind of a wacky way to do it too. How do I remember this one? Before I even say that, I'm going to tell you this is not a perfect analogy, but perhaps it will still help you. We are transformed by the Holy Spirit flowing into us and making us a new creation. Okay? Now, it's not, again, not the same as this because this bacteria dies. The Holy Spirit most certainly is not dead. This bacteria has to die for this creature to take on its its traits. The Holy Spirit doesn't die, but we are still able to. He is still able to breathe new life into us and transform us. And so that's how I remember transformation. Yeah, it's not a perfect analogy. If you come up with a better one, email me and let me know, and I'll put it in my next video. All right, the last one is transduction. And transduction, we're actually going to talk about in a little bit more detail once we start talking about viruses. But transduction is when DNA gets transferred by a virus, and the bacteria then gets infected by this virus. So DNA transferred by infection with a virus. And I'm not going to go into a lot of detail about that because, again, we're going to go into a lot more detail once we talk about viruses. But here's how I'm going to remember this. In the word transduction, you see the word duck. We know that we get infected by viruses and then we end up getting sick with a cold, or we get sick with the flu, or we get sick with, all sorts of different things. And so if somebody standing next to you has a cold and you can tell that they're about to sneeze, you do not want to get sprayed with snot and bacteria and whatever yuck it is that they have. So when they go and they're getting ready to sneeze, all of a sudden you duck. And you duck so that you don't get sprayed with all their snot and get infected by the virus that infects them. Okay? So that's kind of a wacky way to remember it. But transduction, if the bacteria had ducked, maybe he wouldn't have been infected with the virus that then is going to get that viral DNA transferred into him. So we'll use that as an example, and we'll say, okay, well, we'll try that the way it is then. Okay, so you've got 3 methods of reproduction, of genetic variation. You've got conjugation, where we're pouring the DNA from one into the next. We've got transformation, where the bacteria has to die before that gets taken into another bacteria, and then transduction, where it's transformed or it's transmitted by a virus. Okay, so 3 ways of genetic variation, and those all go along with binary fission, which again is the asexual reproduction. It's going to produce identical offspring. These will produce different offspring because we're recombining genetics. The binary fission will produce identical offspring.